Okay, can somebody confirm you can hear me?
I can hear you.
Thank you. Before we begin, are there any questions? All right, today is May 1st. Let me share my screen here. You should screen now. Today is May 1st, and we should be on chapter 4. I think we did start chapter 4. So, for the lab, you should read the unknown project instructions files for Canvas for next week. It looks like we do not have a lab today. Just checking on that. Yeah. So, there is no lab today. So I will not be there other than to check in for the first 15 minutes and ask if there are any questions. So, if you have no questions, there's no reason to log into the lab today. Any questions about that? All right, let's go to chapter 4. I have that we had talked about the selective permeability of cell membranes and other membranes last time. Any questions about any of that? All right. So the plasma membrane does do other things besides, I don't know, keep the cytoplasm inside the cell. And the plasma membrane does have enzymes for ATP production, at least in prokaryote cells. In fact, in prokaryotes, it's on the cell membrane that the major ATP production is made in cells. Uh, we also find the photosynthetic pigments in the photosynthetic bacteria and archaea are in the cell membrane. What happens is there are foldings. Let me see if I can blow this picture up. There are foldings around the cell membrane. And so the cell wall is, right there. The cell membrane here, let me blow this up even further. The cell membrane actually goes here along the cell wall, and then it outfolds, goes there, outfolds. I don't see them here, so let's move over to here. And then it's moving here, and then outfolds, and then outfolds, moves along the cell wall, then outfolds. And these outfoldings are, I don't know, outfoldings of the cell membrane where the photosynthetic pigments are located. And they are given a name, chromatophore or thylakoids is not they're not eukaryote plants, so they don't have a thala, what the heck is that called? Anyways, they're called thylakoids or chromatophores, the foldings of the cell membrane where the photosynthetic pigments are. If you damage the cell membrane, it will cause leakage of the cytoplasm out of the cell, and that will, of course, kill the cell. You can damage the plasma membrane with alcohols, quaternary ammonium compounds, and then the antibiotics of the class polymyxin, like polymyxin B, puts a hole in the cell membrane. Any questions about any of that? All right, the movement of materials across membranes is extremely important to cells. The movement is actually critical for cells. It can happen by two processes. It can be a passive process or it can be an active process. A passive process is where the molecules move down their concentration gradient, meaning if they're higher outside the cell, then they will move passively into the cell down their concentration gradient, moving from an area that has higher concentration to an area that's lower concentration. Of course, in the reverse, if the molecule is higher inside the cell than outside the cell, the molecule will passively move down its concentration gradient and leave the cell. We can have three different ways of passive movement. It can be simple diffusion, and this works with like oxygen and carbon dioxide. Oxygen simply moves into the cell by simple diffusion, and then carbon dioxide moves out of the cell by simple diffusion. You can also have facilitated diffusion, and this happens by a solid moving down its concentration gradient, going through a transport protein, or maybe I should say a channel protein, and moves down its concentration gradient. That's actually the way fructose moves into cells. If you eat a bunch of fruit and has a lot of fructose in it, you have a lot of fructose in your gut, and it will move down its concentration gradient through a protein in your cell membrane. And it is a passive process, doesn't cost the cell any energy. Any question about any of that? And then the third passive movement of material across the cell membrane is by osmosis. Osmosis is when a solvent, like water, moves into the cell if water is higher outside the cell than inside the cell. Of course, if there's a drought condition and there's more water inside the plant, then the water will move by osmosis from inside the cell to outside the cell. Any question about the passive movement of materials? All right, the active transport of material across the cell membrane happens where the molecule moves against its concentration gradient. So when there's less glucose outside the cell than inside the cell will want to move that glucose into the cell. And it can use active process moving the molecule against its concentration gradient. It does require a transport protein in the cell membrane, and it does require the cell to spin one ATP molecule per molecule of glucose that is moved inside the cell. Any question about any of that? All right, let's talk about those in a little more detail. Simple diffusion is just the movement of a solid from an area of high concentration to an area of low concentration, such as when we put a drop of dye inside a beaker of water. The dye is very more highly concentrated in the drop, and the molecules will diffuse throughout the beaker until the concentration gradient is equal in the beaker, and there's no more drop because it's the whole beaker has become a light purple color. Any question about any of that? Like I said, simple diffusion is very important for the cell getting oxygen inside the cell and carbon dioxide leaving the cell. Facilitated diffusion involves a solid moving down its concentration gradient. And this picture is showing you glucose. Glucose isn't the best example. Fructose is a better example because fructose works this way. When you have more molecules of fructose outside the cell than inside the cell, the fructose can enter the cell through a transporter protein. This is not a transport protein because it's not actively transporting the molecule inside the cell. is working like a channel protein, allowing the fructose to move from areas of high concentration to areas of low concentration. That costs the cell no energy. However, that also means that when you eat fructose, not all of it will ever be digested, because once the fructose outside the cell equals the fructose inside the cell, there will be no net movement of fructose either in or out of the cell. So some of the fructose will never get digested. Any questions about any of that? Now, with glucose, it's actually a much more complicated story, and I don't know if I even want to talk to you about it. But with fructose, when there's higher higher glucose concentration outside the cell than inside the cell, the channel protein will allow glucose to move into the cell by facilitated diffusion, costing the cell no energy. But once the glucose outside the cell equals the glucose inside the cell, this channel protein changes from a channel protein acting on facilitated diffusion to an active transport protein. And then all the fructose outside, not fructose, all the glucose outside the cell will be pumped into the cell by active transport. That will cost the cell 1 ATP molecule per molecule of glucose moved into the cell, because the glucose is moving against its concentration gradient. And that means if you eat glucose or any other sugar similar to glucose that's converted into glucose, that all of that sugar will be digested. None of it will remain in the gut. Any question about any of that? So like I said, glucose is a much more complicated story where the protein initially acts as a channel protein, and it allows the glucose to move into the cell by facilitated diffusion. But once the glucose outside the cell equals inside the cell, that channel protein changes to an active transport protein, and then all of the glucose can be pumped into the cell. So with passive processes, both in simple diffusion and facilitated diffusion, solutes are transported down a concentration gradient from areas of high concentration to areas of low concentration. With osmosis, we don't have a solute that's transported, we have a solvent that's transported. And with the term osmosis, that solvent is always water, because water is very important for life. And so when water is moved into the cell, we call it osmosis. And the water always moves from areas of high concentration to areas of low concentration. Now, realize that the tonicity is not defined by the concentration of water. It's defined by the concentration of the solute in the water. And wherever the water is higher, then the solute concentration will be lower. And wherever the water is lower, the solute concentration is higher. Any question about any of that? So a bacterial cell can experience any of three kinds of osmotic solutions. These are called tonicity. There's an isotonic solution, a hypotonic solution, and a hypertonic solution. Each of these terms is relative, so it's correct to say that the solution outside the cell is hypertonic relative to the solution inside the cell. The easiest one to discuss is isotonic, and that means that the concentration of solute outside the cell is equal to the concentration of solute inside the cell. If the cell is in an isotonic solution, then there's no net movement of water into or out of the cell. And the cell is happy because it's in a isotonic solution. It's not losing or gaining water. Now, I did say the word net movement, and that's because water can move from outside the cell to inside the cell. But if one molecule moves into the cell, then there's another molecule of water inside the cell that moves out of the cell. So there's no net movement of water. Any question about any of that? Remember, the tonicity is defined by the solute concentration. So an isotonic one is where the solute concentration is the same outside the cell as inside the cell. A hypotonic solution is where the solute concentration is lower outside the cell than inside the cell. If the solute concentration is lower outside the cell, that means the water concentration outside the cell is higher than inside the cell. So there will be movement of water from outside the cell to inside the cell in a hypotonic solution. If the cell has a cell wall, this is not a problem because the water will move into the cell, expanding the cell membrane up to the cell wall. But once the cell membrane reaches the cell wall, it cannot expand any further because the cell wall limits its expansion. That will prevent any further water from moving outside the cell to inside the cell because there's no room for the water molecule inside the cell because the cell membrane cannot expand. And that will prevent the cell from undergoing osmotic lysis because the cell wall is protecting the cell membrane, preventing more water from moving into the cell. However, if the cell wall is damaged or the cell has no cell wall, then water will continue moving into the cell. The cell membrane will continue to expand until the cell membrane bursts, and then the cytoplasm will leak out of the cell, killing the cell. And that is called osmotic lysis. You put red blood cells, which lack a cell wall, into a beaker of water. Water will move in until the cell membrane pops, killing the cell. If you have a bacteria cell around penicillin G, the penicillin G will get into the peptidoglycan layer, preventing the crosslinks from forming, and then the cell membrane will come out of a little hole in the cell wall where that peptidoglycan crosslink is lacking, where the penicillin G is. The cell membrane will pop out of that hole in the cell wall, and it'll just expand until the cell membrane pops, causing the cell to burst by osmotic lysis. Most bacteria live in a hypotonic solution. Like a bacteria that infects you, it's most likely going to be in a hypotonic solution because we have a little more water than the bacteria. And that's how penicillin G actually damages the bacteria infection or the bacteria cell wall, allowing the water to move into the cell until the bacteria cell membrane pops, causing osmotic lysis. Any question about any of that? All right, a hypertonic solution, the third tonicity that a cell can find itself in is where the solute concentration outside the cell is higher than the solute concentration inside the cell. So there's a net movement of water out of the cell. That will mean the cell membrane will pull away from the cell wall and begin to shrink, shown here and shown in yellow. leading to plasmolysis. This generally will lead to cell death, depending on how sensitive the cell is to water loss. Like E. coli would be very sensitive to water leaving the cell. And after a few hours or maybe a day or a few days, the E. coli cell will die. This is actually why we can keep molasses at room temperature for years. My mother had a bottle of molasses for 20 years, and it wasn't bad. And I actually made molasses candy out of it, fed it to my father, and he loved it, didn't die of that. And then the molasses was 30 years, got to be 30 years or older. We're not sure when my mother bought it. And she said, you know, this molasses is really old. You're not using it, and I'm not using it. I'm going to throw it away. And I said, well, if you haven't used it for 10 years, because that's the last time it was used when I made molasses candy for my father, I said, I suppose we may as well throw it away. So that molasses was 30 years old, and it never went bad because whenever the bacteria fell in it, it was in a hypertonic solution, and then the water left the cell, creating plasmolysis, leading to most of the bacteria dying, even if there was an endospore in the bacteria that fell into the molasses. The endospore was still alive, I'm sure, even after 30 years, but it never germinated, never grew in the molasses because it was always hypertonic. Any question about any of that? All right, so let's talk about active transport. The movement against the concentration gradient is okay when the concentration of substances required by the cell is higher outside the cell than inside the cell. But what about when a cell is in an environment with low nutrient concentrations, such as a cell in the ocean or a cell in the Sahara Desert? Very little nutrients will be found outside the cell compared to inside the cell. And how do these cells survive? By active transport, the movement of material against the concentration gradient. To move a substance or a molecule against its concentration gradient requires the cell to spend energy. It takes work to move the molecule against its concentration gradient. The cell has to have an active transport protein to pump the solute molecule through the cell membrane against its concentration gradient. This costs the cell 1 ATP molecule per molecule moved against its concentration gradient. So the cell has to spend 1 ATP molecule for each molecule it moves into the cell, or against its concentration gradient, because it could pump it out of the cell if it wanted to. All right, any question about active transport? All cells can engage in active transport. There is a form of active transport which is only seen in prokaryote cells. So it's only found in bacteria and archaea. It's called group translocation. It does require a transport protein to pump the molecule across the cell membrane. It does require the cell to spend energy, but the cell usually doesn't use ATP in group translocation. It uses a high energy phosphate compound that's called PEP. You don't really need to know PEP, but you have to use a high energy phosphate compound, which will be used up, meaning broken down, to move the molecule against its concentration gradient. And glucose can move into a bacteria cell by group translocation, but the glucose moving through the transport protein is actually chemically altered. So it starts as glucose outside the cell, but inside the cell, it'll be switched to glucose 6-phosphate, meaning the phosphate from PEP is removed from PEP and then attached to the glucose. It's thought that the phosphate moving on to glucose actually helps the glucose be pumped into the cell and then helps the cell hold on to the glucose so the glucose molecule does not escape the cell, allowing for the accumulation of more glucose inside the cell easier than if the cell was simply using active transport to move the glucose into the cell. Let me explain that a little better if I can get Pate to work here. So here we have our cell, and let's say we have 10 glucose molecules inside the cell, and maybe I can just copy and paste this. And 10 glucose molecules outside the cell. You can move all 10 of these into the cell by active transport. I don't have the arrow here. If we did that on the last one, so let's make this text now. On the last one that moved into the cell, we would be moving one glucose, I'm just going to go G, in against 19 glucose inside. That's quite a stiff gradient difference. One outside, 19 inside. You're pumping the glucose into the cell against a stiff concentration gradient. Need to blow that up a little. Oh, didn't spell that right. Gradient. Let's say we break this up, which is actually something that uh, bacteria cell do. And in this one, both active transport and group translocation are occurring. So here we have 5 glucose move in by active transport. Let's see if I can blow that up a little more. Here we go. And 5 glucose move in by group translocation. Oop. So when we look at the concentration gradient, we'll talk about for the active transport, there's one glucose outside the cell moving in against not 19 now, but 15. Yep. I should have done this by group translocation because it's one glucose inside the cell against four. It's actually glucose 6 phosphate. We'll go G dash P inside. So anyways, the point is that even in the active transport, this is a less steep gradient because instead of one glucose moving against 19, it's now only one glucose moving against 15. And then the group translocation, it's one outside the cell moving inside the cell against 4 glucose 6 phosphate inside the cell. The point is it's much easier moving the glucose from outside the cell to inside the cell if the cell splits it into using both active transport and glucose 6 phosphate. Any question about any of that? Than if the cell only used active transport. No, eukaryotes can only use active transport because they do not have group translocation. But all prokaryotes have group translocation. And to move molecules inside their cell, they will move some by active transport and others by group translocation. All right. So let's talk a little bit about cytoplasm. The cytoplasm is the substance inside the cell membrane. When we're talking about bacteria, all of the stuff inside the cell membrane is the cytoplasm because there is no nucleus. The cytoplasm is mostly water, about 80%. It does have dissolved solutes in it, like dissolved protein, dissolved glucose, dissolved ATP, dissolved amino acids, and obviously dissolved DNA. Wherever you find the DNA in the cytoplasm, we call that the nucleoid region, but there's no boundary between the nucleoid region and the cytoplasm, because wherever the DNA floats in the cytoplasm, that will be the nucleoid region. The nucleoid region does stain differently, but other than that, there is no boundary between the cytoplasm and the nucleoid region. You can also find ribosomes and inclusions in the cytoplasm, but the ribosomes and the inclusions are not dissolved in the cytoplasm. The ribosomes are big enough that they do not dissolve in the cytoplasm, and the inclusions were materials that were being stored by the cell. And so much material, so many molecules came together that the molecules precipitated out of solution in the cytoplasm, forming an inclusion. And there we have an inclusion, the artist's rendition. That might be an inclusion there, that might be an inclusion there. Any question about any of that? So we talked about the nuclear area called the nucleoid region. It's wherever the DNA is, I should say the chromosomal DNA, and there is no boundary between where the DNA is and the cytoplasm. There are also plasmids in a cell. The plasmid is a very small molecule of DNA being independent from the chromosome. Not all bacteria have a plasmid. In fact, some yeast even have plasmids, and that's a eukaryote cell. But some bacteria do have plasmids. I've already talked about the ribosome, so I don't need to talk about it further. Let me talk a little bit about the ribosomes. You do not need to know the subunits of a ribosome, but you should know that ribosomes are made up of subunits. Ribosomes are both protein and RNA. We call it ribosomal protein or ribosomal RNA or RNA. They get together to form the ribosome. The prokaryote ribosomes are 70S in size. That's a little smaller than the eukaryote ribosome, which is 80S in size. The S is how molecular biologists and biochemists measure ribosomes. It actually stands for the sedimentation coefficient in an ultracentrifuge gradient. And when you put the prokaryote ribosomes and the eukaryote ribosomes in an ultracentrifuge gradient and then spin the gradient, the 70S ribosomes go to the 70S size or where the ribosomes sediment in the ultracentrifuge gradient, and the 80S are bigger, and so they sediment down deeper in the ultracentrifuge gradient. You don't need to know anything about the S sedimentation coefficient, but you should know that prokaryote ribosomes are a little smaller than eukaryote ribosomes. Any question about any of that? All right, I mentioned that inclusions are just reserve deposits within the cytoplasm. So much material has been accumulated by the cell that the reserve deposit actually precipitates out of the cytoplasm, meaning precipitates out of solution, coming out of the cytoplasm, forming the inclusion. The inclusions can be used by the cell when resources are low. There are different inclusions. You don't need to know the names. All you need to know is there are different types of inclusions. Here we're looking at some inclusions. Magnetosomes here, the black dots there. This is a sulfur granule. This cell must metabolize sulfur, so the cell is accumulating sulfur inside the cell. And then here, that yellow line, let me blow that up a little, is showing you the cell there, and you can see three inclusions in this cell, the blue circular things. These are metachromatic granules. You don't need to know what that is, but these are some inclusions. Any question about inclusions? All right. Let's blow it up a little more. Another structure we can find internal to some cells is an endospore. Endospores are specialized resting cells produced by certain species, mainly members of the family Bacillus and Clostridium. These endospores are highly resistant to desiccation, heat, chemicals, radiation, and the endospore can survive conditions that the vegetative cell cannot. So whenever the cell is in a deteriorating environment, the cell knows that the environment is going bad and that the cell is not going to be able to survive the environment. And so if the cell is a member of the family Bacillus or Clostridium, it will begin making an endospore. So sporulation or sporogenesis is the forming of the endospore, and it actually is made inside the parent cell. Let me blow this up. So there we see one parent cell here, and it's making an endospore there, that round clear thing there. Here's an endospore here, and there's the parent cell there. Any question about any of that? Oops, didn't mean to do that. Let's get that back. I just wanted to shrink it down to the normal size. There we go. A germination is when the endospore returns to the vegetative cell, where the endospore germinates, giving rise to the parent cell, a vegetative cell. So when the Endospore is in a bad environment, a poor environment. The cell will remain in endospore, but if the environment ever changes and becomes favorable to the species growing and reproducing, the endospore will germinate, return to the vegetative cell. The vegetative cell can grow and reproduce, and it's also metabolically active. I didn't mention it, but the endospore has very low, if any, metabolism going on. So very little metabolism going on in endospore, and that's how the endospore can survive for so long. There are some endospores that they found in like the Egyptian mummies, which are 8,000 years old or older, and they've gotten them to germinate after 8,000 years. The oldest endospore, I think, was something like 40,000 years, and they got that from an endospore they dug out of the Antarctic ice core. And that cell, obviously, endospore was frozen too, but the point is they were able to drill it out of the ice of Antarctica, and it was at least 40,000 years old. They got it to germinate and then form a vegetative cell, which then grew other species. Any question about any of that? All right, let's take a look at endospore formation. Let me blow this up. So endospore formation begins by the cell replicating its DNA. One DNA molecule moves to one side of the cell, the other to the other side of the cell, and then the cell begins dividing. But it doesn't begin dividing as the bacteria cell normally does by leaving a blank there. It normally divides in half, so it normally divides, splitting the cell equally in two parts. Endospore formation, the cell divides with a little cell on one end and a bigger cell on the other end. The bigger cell remains the parent cell that forms the endospore, and the plasma membrane will separate one of the molecules of DNA. It'll actually form two membranes, two cell membranes around this DNA. The cell membrane will then have peptidoglycan layer put inside of it. A spore coat will be put outside the cell membrane and the peptidoglycan layer. And then when the environment decays, the parent cell will die and that will release the endospore. Any question about any of that? I do have a little video, but it's not the best one. Let's go ahead and watch it. Of an endospore forming. It just shows you an endospore forming inside.
Audiences agree Marvel Studios' Thunderbolts is an MCU game changer. It's raw and deeply human.
No one here.
Sorry, a commercial. So this cell is making an endospore. Like I said, not the best video, but it is a video. You should also notice that the cell over here, which was on the furthest to the left, moved a little bit inside the cell. That doesn't always happen. It depends on the species. And you can actually classify the cell by where the endospore ends up in the parent cell. And this one would be a little bit off to the side. It's not in the center. There are some that actually move to the center. There's some that stay on one end of the cell. All right, any questions about endospores? So let's briefly talk about eukaryote cell structure. An animal cell is shown on the right, a plant cell is shown on the left, and you can see that there are many organelles which are found in both animal cells and plant cells, like the nucleus, the Golgi complex, the mitochondria, perioxysomes. There are some organelles or some structures found only in an animal cell and others found only in a plant cell, like the chloroplast is only found in a green photosynthetic plant cell or an algae cell. That's a protist, but we're only going to talk about animals and plants. There's no chloroplast ever found in an animal cell. And remember, there's no chloroplast found in a plant cell that's not green, such as a root cell of the plant. There is a central vacuole found in a plant cell. There are no central vacuoles found in an animal cell. There are vacuoles found in an animal cell and vacuoles found in a plant cell. Unfortunately, I don't have a normal vacuole. That's a central vacuole. A normal vacuole looks very much like a lysosome or a perioxysome. And we'll talk a little bit about that. I'll briefly mention that later in a few minutes. One structure found in a animal cell that is not found in the plant cell is the centrosome composed of a centriole. Blow this up. Oh, that didn't work. So there's the centriole here. The centriole is involved in mitosis, helping to form the mitotic spindle. And it is true the plant cells have a mitotic spindle, but they don't have a centriole. So that's one structure you find in animal cells. This is not a membrane-bound organelle, but it is a cell structure. Any questions about any of that? All right, this is an actual electron microscopic image of an animal cell and a plant cell. No, actually, that's not a plant cell, but an algae cell, which is similar to a plant cell. All right, this slide is showing you some differences between a prokaryotic cell and a eukaryote cell. The difference is in the size. The prokaryote cell is about 1/10 the size of a eukaryote cell, meaning the eukaryote cell is much larger. The eukaryote cell has a true nucleus, no nucleus in a prokaryote cell. Membrane and bound organelles are present in the eukaryote cell. They're absent in the prokaryote cells. There's some difference in the flagella, but I'm not going to talk about it. The glycocalyx is a capsule or a slime layer of a prokaryote cell. There is a glycocalyx in some cells that lack a cell wall, like some animal cells and some protists have a glycocalyx. The cell wall is chemically complex in the prokaryote cell and is usually present, but there are some prokaryote cells that lack a cell wall. And then the cell wall is found on fungi and plant cells and some protists, but it's chemically simple. Like the cell wall of plants is only made of cellulose, much simpler than the cell wall of a prokaryote cell. The plasma membrane is different. It's a little simpler in the prokaryotes, no carbohydrates. It's not entirely true that they all lack sterol, so I won't talk about that. And then the plasma membrane of animal cells do have a sterol in it, and all the plasma membrane of all eukaryotes have a carbohydrates and proteins in it. Of course, the plasma membrane here has proteins in it. Cytoplasm. This is a little bit old. There is a cytoskeleton in the prokaryote cell. It's been found now about 15, 20 years ago. Like I said, this is an old slide and the author never corrected it. There's no cytoplasmic streaming in a prokaryote cell. I'll show you that in a little bit. There is a cytoskeleton, very complex, in a eukaryote cell, and cytoplasmic streaming. The ribosomes differ. The prokaryote ribosomes are a little smaller than the eukaryote ribosomes. The chromosome is usually a single circular chromosome of a bacteria. They lack histone proteins in the chromosome. And the eukaryote chromosome is usually multiple and they're linear, meaning the X chromosome has a beginning place and an ending place. And the chromosome has the DNA wrapped around the histones. So there's histone proteins in the chromosome. The cell, the prokaryote cell divides by binary fission. The eukaryote cell divides by mitosis. Sexual recombination, essentially none, although some bacteria can transfer plasmid DNA, but a sexual recombination happens in most eukaryote cells and it involves meiosis. Any question about any of that? All right, I did mention if the prokaryote cell can move, it moves only with the flagellum. If the eukaryote cell moves, it can either move with flagellum, cilia, or by pseudopods. A flagellum is a long whip-like structure. The cilia are many and usually hair-like structures. They all beat in unison, so the cell will move that way and the cilia move that way on the thing, on the probably a protus here. Pseudopods, I'm not going to show you how it moves. Let's take a look at the flagella here. After the commercial.
The video shows a typical euglenophyte euglena.
There's a
Euglena
flagella
does
there.
not have a rigid cell wall, usually referred to as a pellicle.
The cell is also
the
moving
shape
by
changes as the organism moves.
pseudopods, putting its membrane out there, but it does have flagella as well. All right, I'm not going to talk about the cell wall. You can read about the cell wall. It should be review for you. I'm not going to talk about the cell membrane because I've already talked about it for prokaryote cells. I guess one thing that the eukaryote cell has that the prokaryote cell lacks is eukaryotes can move material across the cell membrane by vesicles called vesicular transport, where a membrane-bound organelle called a transport vesicle brings material to the cell membrane, and the membrane-bound organelle called the vesicle can actually fuse with the cell membrane, and then the vesicle can then dump the molecules inside the vesicle to outside the cell. Like when a teenager has raging hormones, that can happen by the teenager making the hormones in a vesicle, and then the vesicle will fuse with the cell membrane, dumping the hormones outside the cell. two types of vesicle transport: endocytosis and exocytosis. Exocytosis is where the material moves up to the cell membrane and then fuses with the cell membrane, dumping the contents outside the cell. Endocytosis is where material moves into the cell by a vesicle, and this can happen either by phagocytosis or pinocytosis. Phagocytosis is when pseudopods extend from the cell, growing around whatever the cell wants to bring inside the cell, and then the vesicle forms when the pseudopods meet and then fuse, and then that will bring the material inside the cell, called cellular eating or phagocytosis. Pinocytosis is where the membrane invaginates, the molecules being like receptor proteins being on the cell membrane. Once the ligand binds to the receptor, this can cause pinocytosis to happen, where the membrane starts unfolding in on itself. I think I had a picture of this. Nope, I don't. So here's our cell here, and then we have a receptor here, and a ligand binds to this receptor, and there's usually several of them. This membrane will then start unfolding, and the receptor will move down here, and then it'll fold further. And so let's make this, maybe I can cut and paste it right here. Right down here. And then the membrane will fuse. And then the cell membrane will pinch off. And then this vesicle here is now inside the cell, and it can move inside the cell. Any question about any of that? This is showing you a little bit of the cytoskeleton of a eukaryote cell. So the cell, which you can't really see here, is right there. And the cytoskeleton is a framework or backbone of proteins running throughout the cell. Here we have two cytoskeletal proteins, microtubules in green, actin tubules or filaments in red, and the blue is actually the nucleus. Anyways, there you can actually see the cell here. So this would be one cell here, and there's another cell and another cell. So the cytoskeleton made up of three different types of protein filaments. You don't need to know those, but just know the cytoskeleton is a very complex network making the backbone of the eukaryote cell. There is some cytoskeleton in a prokaryote cell, but it's much simpler, much fewer proteins running through the cytoskeleton. I did mention the ribosomes of eukaryotes are larger. They're 80S in size. I think I've mentioned that the eukaryote cells have membrane-bound organelles, or I usually just call it organelles, specialized membrane-bound structures, allowing the cell to be made into compartments, and each compartment can have a specific function by a different membrane-bound organelle. Like the nucleus is a membrane-bound organelle, which serves to hold and protect the DNA. There are some of the different organelles of a plant cell and a animal cell. I'm not going to go through this. Let me briefly talk a little bit about the eukaryote endomembrane system. In eukaryotes, we have an endomembrane system where molecules begin with the ER, endoplasmic reticulum. And if it's a protein molecule, it'll be made on the rough ER. But if it's a lipid, it'll be made on the smooth ER. The rough ER just has ribosomes attached to it. The smooth ER lacks the ribosomes. Let's talk about the rough ER. We can have a protein made on the rough ER, and then the membrane transport endomembrane system involves the rough ER forming a vesicle carrying proteins that were made in the rough ER to the Golgi apparatus. So this is called a transport vesicle, and it's transporting the proteins from the Golgi apparatus, from the ER, the rough ER, to the Golgi apparatus. The same thing happens with lipids, but they come from the smooth ER, forming a transport vesicle, which also moves to the Golgi apparatus. The transport vesicle binds on one end of the Golgi apparatus, and then the molecule, which in this case we're just going to talk about it being a protein, can be modified, chemically altered, and then the protein moves towards this end of the Golgi apparatus. And when the protein is finished, it'll then bud out of the Golgi apparatus, forming another transport vesicle, where the protein will move to where the protein should be, meaning the transport vesicle will transport the proteins to where they should be. In this case, the transport protein was being made to be excreted out of the cell, and the transport vesicle can then fuse with the cell membrane, dumping the proteins outside the cell, which could happen like in a teenager and raging hormones. But the transport protein can go elsewhere, the transport vesicle can go elsewhere. And in this case, the molecules are going here. Whenever the transport vesicle, right here, we're forming a lysosome. But wherever the transport vesicle takes the proteins or whatever to a certain location, and then the transport protein stops moving and the cell just stores it, it's no longer called the transport vesicle because it's not moving. It's called a vacuole, which is simply storing the contents for later use. So a transport vesicle will be the same as a vacuole. It's just the vacuole does not move and it's just storing something. The transport vesicle is moving the contents to a certain location. And initially, a transport vesicle moves the proteins from the rough ER to the Golgi apparatus, but later the Golgi apparatus buds off, forming the transport vesicle, which moves in this case, proteins, to where the protein should be. Any question about any of that? That's the endomembrane system. All right, let's move on to the last one, and that is the mitochondria and the chloroplast differ because they have two membranes around the organelle, and they also have DNA inside the organelle and ribosomes inside the organelle. The endosymbiotic theory is a theory to explain the evolution of chloroplasts in eukaryote cells. How did these organelles originate, and why do they have ribosomes and DNA inside them? Why do they have two cell membranes also? The theory states that a long time ago, we had an ancestral prokaryote cell and an ancestor eukaryote cell that we'll call the proto-eukaryote. And the proto-eukaryote engulfed a ancestor cell. Let me start drawing this now. So here we have the proto-eukaryote. I'm just going to go P E for proto-eukaryote. And the proto-eukaryote can only make ATP by fermentation. I don't think we've talked about that yet, but we will. And it can make one glucose to be made into 2 ATP. Oops. And there's some other things they're made, but we're only going to talk about the ATP here. And that's the only way that this proto-eukaryote could make ATP. Use up one glucose and you get 2 ATP. However, we had a bacteria cell over here. Let me try and draw that. Oops, can't do that there. Let's go ahead and make it right here. So we have a bacteria here, and it can make ATP from aerobic respiration, which we haven't talked about yet, I know, but getting an introduction now. Aerobic respiration, where it could take one glucose and make 38 ATP. A lot more, is the point. Well, the proto-eukaryote engulfed the bacteria by putting out pseudopods. Let me get the eraser here. Growing the pseudopods. Pseudopods met, they then fused together. Bringing the bacteria cell inside the proto-eukaryote. And for whatever reason, the proto-eukaryote did not eat the bacteria right away. So it kept the cell around for later use, probably for later eating, but it didn't use up the bacteria right away. And just by diffusion, some of the glucose from the proto-eukaryote, let me get a thing here. One glucose moved from the proto-eukaryote into the bacteria. And then the bacteria perform aerobic respiration, making 38 ATP, and for whatever reason, shared some of that ATP with the proto-eukaryote. I guess we should go 38 there. We don't know how much it shared, but let's just say something like half of it. So what is that? 19. And we don't know what it was. 19 ATP moved into the proto-eukaryote. And Probably the reason why this was happening was because the protoeukaryote and the bacteria, this is early in life, and the cell wasn't controlling things too well, and so the protoeukaryote allowed the glucose to move by diffusion into the bacteria. And the protoeukaryote said, so what? I'm going to eat the bacteria a little bit anyway, so fine if he gets some of my glucose. And then the bacteria shared some of its ATP, probably because life was early and it just wasn't controlling its ATP production and where it was going, and it just moved into the cytoplasm of the protoeukaryote. And then the protoeukaryote said, "What's going on here? I lost one glucose molecule, and then I got a whole lot of ATP. I could only make 2 ATP from this glucose. Where the heck did I get all of this ATP from?" And the prokaryote came to realize that the bacteria was making this ATP from the glucose and could make a lot more ATP from the glucose than the protoeukaryote could. And so the protoeukaryote kept the bacteria around, feeding it glucose, and then the bacteria made ATP for the protoeukaryote. And the two started living in symbiosis. After a long period of time, you now no longer find the bacteria as a free living cell. You only find it inside the protoeukaryote, which today we call a eukaryote, which has a mitochondria inside of it. And then the bacteria became the protoeukaryote's mitochondria, and then the protoeukaryote we now call a eukaryote because it has a mitochondria inside of it. Any questions about any of that? See where we are here. There we go. For animal cells, this happened one time where they engulfed the bacteria, which evolved after many years into the mitochondria. For plant cells, so it happened twice. Once they engulfed a bacteria, which became the mitochondria, and they engulfed another bacteria, which became the chloroplast. And that's the theory for the evolution of mitochondria and chloroplasts. What evidence do we have for this theory? First, we have some eukaryote cells which actually engulf another cell. This is a paramecium. It engulfed an algae, and then the paramecium feeds the algae, gives it sunlight. The algae makes glucose, like a chloroplast, for the protist. We also have some amoeba which engulf bacteria. The amoeba lack mitochondria, but the amoeba will engulf bacteria, and then the amoeba will feed the bacteria glucose, and then the bacteria will make ATP for the amoeba. So we actually find some protists which are literally doing this today. If we look at the membranes of the mitochondria and the chloroplast, they have two membranes. Let me go back to my paint. And that's the membrane of the vesicle, which normally enclose the bacteria. And then we have the membrane of the cell membrane of the bacteria, meaning the mitochondria and the chloroplast have two membranes, and we expect two membranes if a long time ago they were actually engulfed by a protoeukaryote. So that's one piece of evidence. Oops. Come on. If we look at the DNA of the prokaryote cell, the DNA is usually one circular molecule. The DNA of a eukaryote is a linear molecule. If we look at the DNA of a mitochondria and a chloroplast, it's circular DNA. We don't find any histones in the DNA of a bacteria cell. You do find histones in the DNA of a eukaryote chromosome. There are no histones in the DNA of a mitochondria and the chloroplast. If you look at the first amino acid made in protein synthesis, it's formylmethionine in a bacteria. If you look at the first amino acid made in a protein of a eukaryote cell, it's methionine, a similar amino acid but different. And then we look at the first amino acid made in protein synthesis in mitochondria and chloroplast, it's formylmethionine. If you look at the size of the ribosomes, the prokaryote ribosome is 70S in size, a little smaller than the eukaryote ribosome. We look at the ribosomes inside the mitochondria and the chloroplast, they're 70S in size. If you look at the growth of a prokaryote cell, it's by binary fission. Eukaryote cells, the growth occurs by mitosis. If we look at the growth of the mitochondria and the chloroplast, it happens by binary fission. And then the strongest evidence, if you sequence the DNA of a bacteria, it falls within the domain bacteria. If you sequence the DNA of the eukaryote chromosome, it falls within the domain of the DNA of a eukaryote. If you sequence the DNA of the mitochondria and the chloroplast, where do they fall? They fall within the DNA of the domain bacteria. Any questions about any of that? I can add that they've recently discovered another bacteria that has been engulfed by a eukaryote cell, and it's been living with the eukaryote cell for so long now that the two have been living in symbiosis, and that the bacteria is beginning to form another membrane-bound organelle in the eukaryote cell. I think this is a bacteria that can engage in nitrogen fixation. I don't think I have that written on this slide, so I got to look to see where I've got that. I've got that in one of the term slides, but anyways, the point is it looks like the eukaryote cells, at least some of them, are going to get another membrane-bound organelle, which was formed from a bacteria. It needs many more years of evolution before this will happen. So at the moment, it is a bacteria that is living in symbiosis with the eukaryote cell that is engulfed it, but it is evolving into a membrane-bound organelle, meaning it is no longer a free-living bacteria. It requires the eukaryote cell to live in, and it soon will become, well, evolutionary terms, and terms of evolution, it'll soon become a membrane-bound organelle in the eukaryote cell. Any question about any of that? All right. That's it for Chapter 4. Any questions on Chapter 4? If there's no questions on Chapter 4, I'll end this. And remember, there's no lab. I'll be there at 6:30 for 15 minutes to answer questions. But if there's no questions at 6:45 or after the last question is answered, I will log out. All right. See you.
Will you post this now?
I can, yes.